Okay, let's unpack this. Have you ever wondered what truly makes your body tick or, why a simple pill can have such complex effects? Today, we're taking a deep dive into two incredibly intertwined worlds: proteins, the unsung heroes of your biology and pharmacology, the science of how medicines interact with your living system.
Yeah, and what's truly fascinating here is how these two areas, which seem, well, distinct, are profoundly connected at a molecular level. Our mission for you today is really to extract the most important nuggets of knowledge from a stack of sources, giving you a comprehensive understanding of the building blocks of life and the intricate world of drugs. Get ready for some genuine aha moments that I think will change how you look at your body and, yeah, your medicine cabinet.
Think of it as a shortcut to being well informed, maybe with surprising facts, and just enough humor to keep you hooked. So let's kick things off with proteins, the body's master builders.
Yeah.
When we talk about proteins, what are we really talking about? We're talking about one of the most abundant macromolecules in your cells. These are essentially very large, complex molecules, essential for life. And they vary incredibly widely in size and function, doing everything from like building your hair to making your muscles move.
That's a great way to put it. Um, at their most fundamental level, proteins are polymers, meaning they're long chains made up of individual building blocks called amino acids. These amino acids are linked together by strong chemical bonds called peptide bonds. Think of them like sturdy knots holding beads together on a string.
Okay, peptide bonds. And these strings, they can be classified in a couple of ways, right? You mentioned simple proteins first.
Exactly. Simple proteins, or just that made only of amino acids. Classic examples are albumin, you find it in your blood and egg whites, or globulins, which are essential for antibodies and blood clotting, like fibrinogen. And then you also have the structural ones, the scleroproteins like collagen in your skin, and keratin that makes up your hair and nails.
Okay, so just amino acids, but then there are others.
Right. Not all proteins are just amino acids. Some are conjugated proteins. This means they have the amino acid chain plus other non-amino acid components, which we call prosthetic groups. These extra groups are like specialized attachments. They give the protein unique functions.
Ah, so prosthetic groups give them extra powers, sort of. So depending on what those attachments are, we get different types. Like,
Precisely. You get some interesting categories. For instance, lipoproteins have lipids, or fats, attached. And think of the ones that transport cholesterol. Glycoproteins have carbohydrates attached. Many of your antibodies are actually glycoproteins. Then there are phosphoproteins with phosphate groups. Caseins in milk are a great example, making up about 80% of milk protein, vital for phosphorus and calcium delivery. And finally, you've got metalloproteins, which incorporate metals like iron and ferritin for storage, zinc in certain enzymes, or calcium in regulatory proteins like calmodulin. It's a highly diverse family.
wow, okay, so it's not just a simple string.
Not at all. And this diversity isn't accidental. Proteins also have this fascinating hierarchy of structure, from simple to incredibly complex. And this three-dimensional shape is absolutely crucial for their function. It's like intricate biological origami.
Biological origami, I like that. So what's the very first fold in that molecular origami?
Well, the initial level is the primary structure. This is simply the unique linear sequence of amino acids linked via those peptide bonds we mentioned. Think of it as the specific order of beads on that string of beads. It's the most basic and, honestly, in many ways, the most important level. Because this precise sequence dictates every subsequent fold and, ultimately, the protein's function.
Okay, the sequence is key, and then those strings start to fold in predictable patterns, right?
Precisely. The next level is secondary structure. This refers to the localized, regular arrangements of the polypeptide chain. These folds are mainly stabilized by hydrogen bonds. The two most common forms you'll hear about are the alpha helix, which literally looks like a coiled spring.
Like a spiral staircase.
Exactly. And the beta-pleated sheet, which resembles a folded piece of paper, kind of pleated back and forth. You also have specialized forms, like the collagen helix that gives collagen its unique properties, its strength and flexibility.
Okay, helix and sheet. And from there, it gets even more intricately folded.
Yes, absolutely. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. This is the full form of the protein, if you will. It's stabilized by various interactions, electrostatic forces, hydrophobic interactions pushing watery bits away, other intermolecular bonds. It's how the entire string folds up into a compact functional unit.
Okay, so one whole chain folded up.
Right, and then sometimes multiple of these uniquely folded chains come together. That gives you the quaternary structure. This is the arrangement of multiple polypeptide chains or subunits, forming a larger functional protein complex. A perfect example is hemoglobin.
Ah, the oxygen carrier.
That's the one. It's made up of 4 polypeptide chains working together to transport oxygen effectively.
Got it. And speaking of overall shape, I remember there's a basic distinction between two broad types. You have the fibrous proteins.
Correct. Fibrous proteins are typically long, stable, and insoluble. They often serve structural roles. Think keratin in your hair or collagen, giving structure to your skin and bones. Then you have globular proteins. These tend to be more compact, spherical, and more soluble in water. They often act as enzymes or antibodies. They each have a distinct look and purpose, perfectly suited to their jobs.
Okay, that makes sense. Fibrous for structure, globular for action, kind of.
Yeah, in a way. And if we connect this to the bigger picture, proteins aren't just structural components. They perform this incredible array of functions vital for life. One of their most critical roles is as enzymes.
The catalysts, right, speeding things up.
Exactly. They're the biological catalysts enabling nearly all chemical reactions in your cells, from digestion to energy production. And they're highly specialized, like lipases breaking down fats or proteases breaking down other proteins.
You mentioned an example earlier, COX.
Yes, COX, cyclooxygenase, is a great example. This enzyme converts a specific fatty acid, arachidonic acid, into compounds called prostaglandins, which cause pain and inflammation. And here's where medicines tie in. NSAIDs, like aspirin or ibuprofen, work precisely by inhibiting that COX enzyme.
Ah, so they block the enzyme, block the pain signal?
Pretty much. It reduces those inflammatory responses. It's a perfect illustration of how understanding protein function leads directly to effective treatments.
That's cool. What about transport? You mentioned hemoglobin.
Right. Proteins are indispensable for transport. They bind to specific ions or molecules and move them around. Albumin, for instance, is the most abundant protein in your blood plasma. It does a lot, helps balance water, transports poorly soluble things like bilirubin, fatty acids, calcium, hormones, and crucially, even drugs.
So albumin is like a taxi service in the blood?
Kind of, yeah. And of course, hemoglobin transports oxygen in red blood cells. And you have glucose transporters, GLUTs and SGLTs. These help glucose get into your cells or co-transport it with sodium in your gut and kidneys.
They also have a storage function, right? Like ferritin storing iron.
Exactly. Ferritin stores iron, mainly in the liver, keeping a reserve ready for when your body needs it. And movement. Without proteins, we wouldn't be moving much at all. Actin and myosin are the key contractile proteins.
In muscles?
Yep. Essential for muscle contraction, but also for cell movement in general. Blinking an eye, running a marathon, it relies on these proteins working together.
And the structural ones, again, keratin and collagen.
Yeah, on the structural side, proteins like keratin in hair and nails, and collagen, the major component of connective tissues like skin, bone, tendons, they provide support, protection, resistance. It's actually amazing to think that your hair's stretchability and how permanent waves work in hairdressing. That's directly related to keratin structure, its alpha helix shape, how it can shift to a beta conformation, and the role of its strong disulfide bonds.
Wow. And defense. You mentioned antibodies.
Absolutely. Proteins are frontline defenders. Vital for defense, recognizing foreign substances. Antibodies are prime examples. When we talk about food allergies, for instance, most food allergens are actually specific types of proteins, usually glycoproteins.
So cooking can change them.
It can. Cooking or enzymatic breakdown, like digestion, can alter the protein's shape, modifying those allergenic epitopes, the parts the immune system recognizes. Sometimes that reduces the allergic reaction, sometimes not.
Interesting. What else? Regulatory functions.
Oh, yes. Many hormones are proteins or peptides. Think insulin-regulating blood sugar or oxytocin. They perform crucial regulatory functions. Proteins called G proteins are also key signal transmitters inside cells. And in the broader context of maintaining balance, homeostasis, proteins are involved. They help maintain water distribution, and they're crucial for blood coagulation players like fibrinogen and thrombin.
Right, stopping bleeding.
Exactly. They even act as pH buffers, helping to keep the pH levels in your body stable, which is critical. And we can't forget their role as mediators of inflammation. Things like cytokines, TNF-alpha, IL-1 beta, IL-6, these are protein messengers that signal distress and coordinate the body's immune and inflammatory responses.
Okay, so they do basically everything. But what happens when these incredible protein structures go wrong?
That's where problems arise. Sometimes proteins can lose their specific three-dimensional structure, even if the amino acid sequence, the primary structure, remains intact. This is called denaturation. It's often caused by things like heat.
Like cooking an egg.
Exactly like cooking an egg white. It changes permanently. Strong chemicals like urea or detergents or organic solvents can also cause denaturation, and it's generally irreversible.
And sometimes it's not just losing shape, but misfolding. You mentioned prions?
Yes, prions are a dramatic and scary example. These are essentially misfolded proteins that act as infectious agents. They cause rare but devastating degenerative brain diseases like bovine spongiform, encephalopathy, mad cow disease, or Creutzfeldt-Jakob disease in humans. It's a stark reminder of how critical proper protein folding is. A single type of protein misfolding can trigger a cascade, converting normal proteins into the misfolded form.
That's chilling, but proteins can also tell us when something's wrong, right? As markers.
Absolutely. They can serve as crucial markers for disease. For instance, in kidney dysfunction. Finding protein in the urine is a sign of trouble, but the type and size of the protein gives clues about the specific problem. Large proteins like IgG suggest one type of damage, medium ones like albumin another, and small ones like microglobulins yet another kind. It helps doctors pinpoint the issue.
Okay. Any other examples?
Sure. Alpha-1 antitrypsin, or AAT, is a protease inhibitor if you have a deficiency.
So it normally stops enzymes from breaking things down.
Right. If you lack AAT, unchecked enzymes, especially one called elastase, can attack the elastin in your lungs. This loss of elasticity leads to pulmonary emphysema. Levels of another one, alpha-1 acid glycoprotein, or AG, can also tell us a lot. Increased levels might indicate inflammation, like in rheumatoid arthritis, certain cancers, burns, or even a heart attack.
And low levels.
Decreased levels can signal malnutrition or severe liver disease. And then there's C-reactive proteins, CRP. It's a widely used nonspecific marker. Synthesized in the liver, its levels shoot up in response to infection, tissue injury, or inflammation.
So it tells you something is wrong, but not exactly what.
Precisely. It's an indicator of inflammation somewhere in the body.
Let's circle back to hemoglobin and oxygen transport. You said other things affect how well it works?
Yes, it's a great example of protein function being finely tuned. The efficiency of oxygen transport isn't just about how much hemoglobin you have. It's also influenced by factors like pH. Lower pH, more acidic conditions actually makes hemoglobin release oxygen more easily. This is known as the Bohr effect.
Useful in tissues that are working hard in producing acid.
Exactly. Also, carbon dioxide levels and a molecule called 2-3-bisphosphoglycerate, or 2,3-BPG, influence how tightly hemoglobin holds onto oxygen. They all modulate its affinity.
And things can go wrong with hemoglobin binding too.
Definitely. You can have carboxyhemoglobin, where hemoglobin binds to carbon monoxide, which does much more strongly than oxygen. This drastically reduces oxygen transport. That's why CO poisoning is so dangerous. Or methemoglobinemia, where the iron in hemoglobin gets oxidized to the wrong state, F3+ instead of F2+ N, and can't bind oxygen properly. And a classic genetic example of protein behaving badly is sickle cell anemia, anemia falciforme.
Right, the shape changes.
Yeah, a single mutation in the gene for one of the hemoglobin chains leads to an abnormal hemoglobin molecule. This causes red blood cells to become stiff and sickle-shaped, especially under low oxygen conditions. This reduces oxygen transport, and worse, these stiff cells can block small blood vessels, causing pain and organ damage.
And just the overall amount of protein in the blood can be an indicator.
Yes, general protein levels can reflect overall health or hydration status. Hyperproteinemia, high protein levels, often points to dehydration.
Not enough water, so the protein concentration goes up.
Basically, yes. Either from inadequate fluid intake or excessive fluid loss, think vomiting or severe diarrhea. Conversely, hypoproteinemia, low protein levels, can have several causes. It could be increased plasma volume diluting the protein, or actual protein loss through the kidneys, like in glomerulonephritis, or through the skin from severe burns, or it could simply be a diet severely lacking in protein.
Okay, so we've covered structure, function, what happens when things go wrong. What about the life cycle, making and breaking down proteins?
Right, the dynamics. Protein biosynthesis is this incredibly complex process, essentially translating genetic information. It starts with the DNA blueprint, which gets transcribed into messenger RNA, mRNA. Then ribosomes, which contain ribosomal RNA, rRNA, read the mRNA code. And transfer RNA, tRNA, brings the correct amino acids to the ribosome to be linked together into the polypeptide chain. It's a cellular assembly line.
A very intricate one. Now, you raised an interesting question earlier. Could a diet of only proteins meet energy needs?
Yes.
Yeah.
Theoretically.
Yeah.
It could, for a while at least. It comes down to gluconeogenesis. Certain amino acids, called gluconeogenic amino acids, can be converted by the body into intermediates, like pyruvate or oxaloacetate. These intermediates can then enter the gluconeogenesis pathway, which is essentially the process of making new glucose from non-carbohydrate sources.
So the body can make sugar from protein if it has to.
It can. It's a remarkable backup system. Though relying solely on protein isn't ideal for balanced nutrition or long-term health for various reasons.
Okay. So what does this all mean? We've seen how proteins are just fundamental, the very essence of life. From regulating our pH to forming our hair, they're built with incredible precision, and even a tiny mistake in their structure can have huge consequences. But just as our bodies are complex, so too are the substances we deliberately introduce into them. Let's shift gears now to pharmacology. This is where understanding the dose can literally mean the difference between a remedy and, well, a poison.
Indeed. This is exactly where the intricacies of biology meet the practical application of chemistry, and knowing how they interact is absolutely crucial for health and safety.
The word pharmacology itself kind of tells a story, doesn't it? From the Greek, pharmakon meaning both drug and poison, and logos meaning study, it really sets the stage for the delicate balance involved.
It truly does. And there's that famous quote from Paracelsus, the physician from centuries ago.
The dose makes the poison.
Essentially, yes. He said, "All substances are poisons. There is none which is not a poison. The right dose differentiates a remedy from a poison." This is really a cornerstone of pharmacology. It reminds us that even beneficial substances need incredibly careful handling.
That's powerful. And just to clarify terms, when we talk about a drug, what exactly is it compared to a remedy or a medicament?
Good question. The nuances matter. A drug usually refers to a substance with a medicinal or sanitary purpose, often the raw active ingredient. A remedy is broader. It's anything that cures or alleviates a disease. Could be a drug, could be rest, could be chicken soup. A medicament or medicine is specifically a pharmaceutical product, technically prepared and formulated for prophylactic, curative, palliative, or diagnostic purposes.
Okay, so every medicine is a remedy, but not vice versa.
Exactly. And what's fascinating here and really important is that a medicine isn't just any merchandise. It should be used with caution by a patient, not just treated like a consumer product. It highlights the responsibility involved in its use, its special status, compared to everyday goods.
Yeah, that's a key distinction, especially when you think about the line between food or supplements and medicines. Foods can't just make health claims, right?
Correct. They cannot make unproven health or therapeutic claims. The vitamin C example is perfect. If you're taking it within the daily recommended dietary allowance, RDA or IDR as it's sometimes called, it's generally considered a supplement. But if you take doses significantly outside that range, say well above 100% of the RDA, it crosses into the territory of being a medicine because it's exerting potential pharmacological effects beyond basic nutrition. The dose matters.
Right, always back to the dose. So when we talk about how medicines work, what are the key concepts? Dose and...
Dose and half-life are fundamental. The dose, as we've said, is the quantity administered, often based on patient weight or other factors. The half-life is the time it takes for the concentration of the drug in your blood plasma to decrease by 50%.
Why is that important?
Because it dictates how long the drug stays active in your system and how often you need to take it to maintain an effective level. Penicillin, for example, has a very short half-life, maybe 30 minutes. Paracetamol, or acetaminophen, is longer, maybe 1 to 4 hours. So the dosing interval, how many times a day you take it, is directly related to its half-life.
Okay, dose and half-life. But just getting the dose right isn't enough, is it? It has to actually work and be safe.
Absolutely. That's the core principle.
Right.
A drug must be both efficacious, it works, and safe. We often measure this balance using the therapeutic index, IT, or therapeutic window. That's basically a ratio. It compares the dose that causes toxicity in 50% of the population, the LD50 or lethal dose 50, to the dose that is effective in 50% of the population, the ED50 or effective dose 50.
So a higher number is better, safer.
Yeah, exactly. A higher index means a wider margin between an effective dose and a toxic one, making the drug generally safer. Some drugs, like digoxin for heart conditions or valproic acid for seizures, have a low therapeutic index. The effective and toxic doses are close together, requiring careful monitoring. Others, like amitriptyline or paracetamol, have a much higher, wider index, meaning there's more room for error, though toxicity is still possible at high doses.
Got it. What about bioavailability?
Ah, yes. Biodisponibility or bioavailability. That's the percentage or fraction of the administered drug dose that actually reaches the systemic circulation, the bloodstream, unchanged.
So not everything you swallow gets used.
Not necessarily, especially for oral drugs. Some might be broken down in the gut or liver before reaching the blood. 4V drugs, by definition, have 100% bioavailability because they go straight into the vein. And related to this is bioequivalentia or bioequivalent.
It sounds important for generic drugs.
Extremely important. Bioequivalence means that two medicines containing the same active ingredient in the same strength and dosage form given by the same route show no significant difference in their rate and extent of absorption, essentially. They have identical bioavailabilities. This is the scientific basis for allowing generic drugs to substitute for brand name reference medicines. They're proven to work the same way in your body.
Okay, that makes sense. When you get a medicine, there's always that folded up paper inside the package insert.
Yes, the BROLAs or package inserts. These are actually legal documents. They provide crucial information for the safe and effective use of the medicine. Often there are two versions, one written in more accessible terms for patients and a more technical one intended for healthcare professionals.
And the box itself.
The packaging is also important. We distinguish between primary packaging, the stuff in direct contact with the medicine, like blister packs, vials, ampoules, strips, tubes,
Like the immediate container.
and secondary packaging, which is the outer box or carton that holds the primary pack and usually contains the insert.
And inside the pill or liquid, it's not just the active drug, right? There's other stuff.
Correct. Besides the active pharmaceutical ingredient, API, medicines contain excipients. These are inactive ingredients. They don't have a therapeutic effect themselves, but they're crucial. They help formulate the medicine, give tablets bulk, help them dissolve. They protect the drug, improve stability, control release, aid manufacturing, even make it taste better.
So they're like the supporting cast for the star active ingredient.
That's a good analogy, yes. They're essential for making the medicine practical and effective.
Which probably explains why drugs come in so many different pharmaceutical forms: tablets, capsules, syrups, creams, injections.
Exactly. It's not just about convenience, although that's part of it. The form is chosen for specific reasons.
Yeah.
Ease of administration, achieving precise dosing, protecting the drug from things like stomach acid or light, ensuring it gets absorbed properly, or targeting delivery to a specific site like skin cream.
Makes sense. You wouldn't want to swallow a skin cream.
No. Precisely. We see solid forms like tablets, capsules, powders, suppositories, even adhesive patches, liquid forms like solutions, syrups, suspensions, elixirs, emulsions, shampoos, and semi-solid forms like creams, ointments, gels, pastes.
And some tablets are designed differently, like slow release.
Yes, you have different release forms. Immediate release is standard. Prolonged or sustained release delivers the drug slowly over time, and retarded or delayed release, like enteric-coated tablets, are designed to pass through the stomach intact and release the drug in the intestine, protecting either the drug from acid or the stomach from the drug.
Okay. How else are medicines classified? By what's in them?
Yes, by composition. You have simple medicines, which contain only one active drug, like simethicone for gas relief. Then there are compound medicines or combination products, which contain two or more active drugs. Think of many multi-symptom cold and flu remedies.
And within the market, there are different types, like the original brand versus generics.
Right. There's a key hierarchy. The reference medicine is the innovator product, the first one approved and marketed, usually by the company that developed it, after proving its efficacy and safety through extensive trials. It sets the standard. A generic medicine contains the same active ingredient in the same dosage form and strength, and is proven to be bioequivalent to the reference medicine. Because it's bioequivalent, it's considered interchangeable.
So it should work exactly the same.
Theoretically, yes. Then there's the similar medicine. This one has the same active ingredient, concentration, form, administration route, dosage, and therapeutic indication as the reference drug, but this is key, it has not undergone bioequivalence testing to prove it acts identically in the body.
So it might be slightly different in how it's absorbed or acts.
It could be. That's why generics are generally preferred for substitution over similars, where bioequivalence is proven. Regulatory bodies, like Anvisa in Brazil or the FDA in the US, maintain lists specifying which generics are deemed equivalent to which reference drugs.
Okay. Are there other special categories?
Yeah, a few worth mentioning. Danamizados are preparations used in therapies like homeopathy, anthroposophy, or homotoxicology, made through specific processes of serial dilution and succussion. Fractionated medicines are sometimes available, where the pharmacy dispenses only the exact quantity needed for the treatment course, rather than a full standard pack, aiming to reduce waste and cost.
Like getting just 7 pills instead of a box of 30.
Exactly. Phytocarapicos, or phytotherapeutics, are medicines obtained exclusively using active ingredients derived from medicinal plants or vegetable raw materials. And a big category is MIPs, medicamentos y centros de prescripción, or over-the-counter OTC drugs. These are vente libre, sold without needing a prescription.
Like pain relievers, cough drops.
Right. Generally, they have a good safety profile when used as directed, fewer contraindications, and are intended for treating minor self-limiting conditions like mild pain, fever, or allergies.
Okay, so OTCs are without prescription. Then we move into the prescription-only medicines.
Correct. These require authorization from a doctor or sometimes a dentist. Within this, there are levels. Some are venda sob prescription, sale under prescription, and might have a red stripe, tarja vermelha, on the box in some countries. The prescription is needed, but the pharmacy doesn't usually keep it. Others also require prescription but do require retention by the pharmacy. These often have stricter warnings like a black stripe tarja preta, or sometimes a red stripe with retention specified. This applies to drugs with a higher potential for dependence, abuse, or serious side effects.
And then there are the really tightly controlled ones.
Yes, the controlled substances, often governed by specific regulations like Portaria 3441998 in Brazil. These are rigorously classified into lists like A, B, C, D, E, F, based on their nature and potential for abuse. We're talking narcotics, list A, psychotropics, list B, retinoids, immunosuppressants, list C, precursors, list D, prohibited substances, list F, and others.
And they need special prescriptions.
They do. Different colored prescription forms, yellow, blue, special white forms, are used for different lists, each with strict rules about validity periods, maximum quantities that can be dispensed, and prescriber registration. It's a system designed for tight oversight of potentially dangerous drugs.
Wow, complex. What about antibiotics? Are they separate?
They often fall under specific rules too, aimed at controlling their use and combating antimicrobial resistance. For instance, RDC 2020L11 in Brazil established specific rules for antimicrobials. They require a prescription, which must be retained by the pharmacy, usually a second copy, but the validity periods and quantity rules might differ from the Portaria 344 controlled substances. It's another layer of control specifically for antibiotics.
So ensuring safe use involves all these regulations, but also the prescription itself, right?
Absolutely. The quality of the prescription is paramount for safety. A few key things. It must have legible handwriting. Illegible scripts are a major source of errors.
That seems obvious, but...
You'd be surprised. Also, avoid ambiguous abbreviations. Write out via oral, by mouth, not just via. Write Acada 4 horas every 4 hours, not 44 hour. Specify exact quantities, 30 capsules, not just one box, because box sizes vary. Physicians are ethically and often legally required to write legibly and clearly. And generally, except in emergencies, prescriptions shouldn't be given over the phone or via mass media, too much risk of error or misunderstanding.
Okay, now here's something you hear a lot. If it's natural, it's safe, or it has no contraindications, here's where it gets really interesting. This common belief, it's a dangerous misconception, isn't it?
Absolutely. It's one of the biggest myths out there. Natural does not automatically mean safe. Many potent poisons are perfectly natural. And crucially, natural products can interact with conventional medicines. A powerful example involves warfarin, a common anticoagulant or blood thinner.
Right, prevents clots.
Yes. If someone taking warfarin also consumes certain common herbs like ginkgo biloba, or even large amounts of garlic, Allium sativum, both of which also have blood-thinning properties, it can significantly increase the risk of bleeding, potentially becoming very dangerous. Another example, some antidepressants can interact badly with Panax ginseng, potentially leading to symptoms like mania.
Wow. So you really need to tell your doctor everything you're taking. Herbs, supplements, everything.
Everything. Natural substances can have potent pharmacological effects and interact in complex ways. Transparency is key for safety.
When drugs don't behave as intended or cause problems, we talk about adverse reactions.
Yes, adverse drug reactions or ADRs. The formal definition is a harmful and unintended response to a drug that occurs at doses normally used for prophylaxis, diagnosis, or therapy. It's useful to differentiate general side effects, which are any effect other than the intended therapeutic one. Some might even be beneficial, from adverse effects, which are specifically undesired, unpleasant, or harmful side effects.
Okay. And sometimes drugs are used for things they weren't originally approved for.
That's called off-label use. It's when a drug is prescribed for a condition, age group, dose, or route of administration that hasn't been specifically approved by the regulatory bodies, like the FDA or EMA, It's often done when there's scientific evidence suggesting potential benefit, but the manufacturer hasn't sought formal approval for that specific use. Sometimes an effect desired emerges that wasn't the primary target, like sildenafil, initially developed for heart conditions, pulmonary hypertension, but famously found to be effective for erectile dysfunction.
What about drug allergies? How do they happen?
Drug allergies are a specific type of ADR involving the immune system. Most drugs themselves, or their breakdown products, metabolites,
Mm.
are actually too small to trigger an immune response directly. They act as haptens, meaning they bind to larger molecules in the body, usually proteins, to form a complex that the immune system then recognizes as foreign or dangerous. This triggers an immune response. There are different types of allergic reactions classified I through 4. Type 1 is the immediate, potentially life-threatening anaphylactic shock. Type 2 involves cells being destroyed, cytotoxic reactions. Type 3 involves immune complexes getting deposited in tissues. And type 4 is delayed hypersensitivity, often seen as contact dermatitis or other skin rashes.
Okay. And what about self-medication? People just taking things without a doctor?
Automedication or self-medication is complex. The World Health Organization actually recognizes responsible self-medication as potentially beneficial for treating minor, well-recognized ailments, potentially saving healthcare resources. However, many factors, lack of clear information, persuasive marketing, seeking a quick fix, contribute to potentially irresponsible self-medication.
What kinds of drugs are common here?
Common classes involved are things like anti-flu or cold remedies,
Right.
NSAIDs for pain, antihistamines for allergies, and worryingly, sometimes even leftover antibiotics, which contributes to resistance.
And the flip side almost, the placebo effect.
Ah, the placebo effect. Fascinating stuff. It's a measurable, observable, or felt improvement in health not attributable to an actual treatment. Essentially, a non-active substance or procedure, the placebo, can produce desired or even sometimes undesired nocebo effects simply because the patient believes or expects it will. It really highlights the powerful connection between the mind and body in healing.
Okay, so back to practicalities. What does this all mean for your daily routine, like taking meds with food or drink?
Oh, this is a huge area. Drug-food and drug-liquid interactions. It can significantly affect how a drug works. Food, especially solid food, can delay gastric emptying how quickly stuff leaves your stomach. It can also alter the stomach's pH. Both of these can directly affect how much of a drug is absorbed and how quickly.
So food usually slows things down. Often, yes. For instance, the absorption of the vitamin riboflavin changes depending on whether you take it with food. Aspirin absorption is delayed by food. Liquids can also influence absorption based on things like their caloric content or pH. For example, acidic drinks like Coca-Cola or grape juice can actually increase the absorption of the antifungal etraconazole. Large volumes of water might affect the action of some antibiotics by diluting them in the gut.
So it's not just about taking it with something, but the right something, or maybe nothing.
Exactly. There are specific recommendations for many drugs. Fatty foods can enhance the absorption of certain fat-soluble drugs, like grisofulvin. Milk or calcium-rich foods can interfere with antibiotics like tetracyclines by binding to them, reducing their absorption.
Oh, right. I've heard that one.
Anti-inflammatories, NSAIDs, are often recommended to be taken with meals, not because it helps absorption, but to reduce stomach irritation, the general rule of thumb.
Oh.
Always prefer taking medication with a full glass of plain water, unless your doctor or pharmacist specifically advises otherwise, like taking it with food or milk.
And what about alcohol? That seems like a bad mix generally.
Usually is. Drug-alcohol interactions can be significant and dangerous. Alcohol can intensify the side effects of many drugs, particularly drowsiness caused by antihistamines, tranquilizers, or sleeping pills. It can cause dangerous spikes in blood pressure with certain antidepressants, MAO inhibitors. With antidiabetics, it can lead to severe hypoglycemia, low blood sugar. It can increase the liver toxicity of drugs like paracetamol. Best to avoid alcohol when taking most medications.
Makes perfect sense. And for those who drive, some meds can be risky.
Very risky. Driving under medication is a serious concern. Many medications, tranquilizers, sedatives, some strong painkillers, many antihistamines, especially older ones, some antidepressants, even some cold medicines can impair your motor skills, coordination, reaction time, visual and auditory perception, and judgment.
So you might not be safe to drive even if you feel okay.
Exactly. The effects can be subtle. Always check the medication label for warnings about driving or operating machinery. And if you're unsure, ask your doctor or pharmacist. Don't take chances.
Okay. What about orphan drugs? What does that mean?
Orphan drugs are medicines developed specifically to treat rare diseases or conditions. They're called orphan because under normal market conditions, the small number of potential patients makes them commercially unattractive for pharmaceutical companies to develop. There's little prospect of recouping the huge R&D costs.
So they need special incentives.
Yes, governments often provide incentives like extended market exclusivity, tax credits, or grants to encourage companies to develop these necessary treatments for patients with rare, often very serious or life-threatening diseases.
That leads to a bigger question. Do medicines always actually solve the problem, achieve the desired result?
That's a crucial point. Not always. In fact, it's important to remember that probably 99% of medicines are, in a way, strange or foreign substances to the body. They're capable of causing numerous side effects, some minor, some severe. And many medicines don't offer a cure, but rather relief of symptoms, management of a chronic condition, or comfort. Success really relies on a chain of events. An accurate diagnosis in the first place, choosing the correct medicine for that diagnosis, prescribing the proper dose, selecting a suitable administration route, and ensuring the timing and duration are right.
If any link in that chain is weak,
The outcome might be less than ideal, or the medicine might not work at all for that particular individual, or could even cause harm. It's not a magic bullet situation.
and unfortunately, errors can creep into this complex system.
Yes, medication errors are a significant concern. They can happen at any stage: prescribing, dispensing, administration. Simple things like those illegible prescriptions we mentioned are a common source, despite ethical requirements for clarity. We also have to consider the influence of the pharmaceutical industry. Their spending on marketing and advertising often significantly exceeds their spending on research and development.
Which can influence prescribing.
It can potentially influence prescribing habits. There's also the phenomenon sometimes called empirotherapia, a term used in some places for practices where pharmacists might be incentivized, formally or informally, to push or recommend certain drugs over others, which might not always be in the patient's best interest. It contributes to improper use or overprescription.
Lastly, a really critical area, pregnancy and breastfeeding.
Absolutely critical. Using drugs in pregnancy and lactation requires extreme caution. There are several considerations. First, the drug's known pharmacological effects might directly impact the fetus, for example, androgenic hormones causing masculinization, or oral anticoagulants causing bleeding risk. Second, and perhaps more worryingly, is the potential for teratogenicity.
Causing birth defects.
Yes. Teratogens are substances that can cause structural or functional alterations in the developing fetus. Infamous examples include thalidomide causing limb defects, misoprostol, certain retinoids used for acne. The stage of fetal development when the drug is taken is crucial. The first trimester is often the most vulnerable period for major structural defects. We also have to consider how easily the drug crosses the placenta to reach the fetus. And finally, drugs can be secreted into breast milk, potentially affecting the nursing infant.
So basically, extreme caution and always consult a doctor.
Always. The risks versus benefits must be carefully weighed for every medication during pregnancy and lactation.
Wow, what a deep dive indeed. We've navigated the microscopic world of proteins, discovered their amazing versatility, how they build and run things, and then plunged into the really complex landscape of pharmacology, learning how these chemical substances interact with our intricate biological systems, with all those proteins. It really makes you appreciate the incredible precision that goes into how our bodies work and, well, how medicines are designed and hopefully used safely.
Indeed. And knowledge, as they say, is most valuable when it's understood and applied. What's truly fascinating here, I think, is the ongoing frontier. Given our increasingly sophisticated understanding of protein structures, thanks to things like structural biology and proteomics, and our deeper knowledge of the molecular mechanisms of disease, how might future therapeutic approaches leverage this knowledge? Could we create truly personalized medicines drugs tailored not just to a disease, but to an individual's unique protein landscape, their specific genetic makeup influencing those proteins? Could we achieve unprecedented efficacy and, crucially, minimize those side effects we talked about?
That's a really provocative thought for you, our listeners, to mull over. Imagine a world where every treatment is perfectly customized, almost like a bespoke suit for your own unique biology. What new possibilities could that unlock? Until next time, stay curious and keep diving deep.